Ever watch those sci-fi movies? You know, where they can fix someone's superpowers or even like completely change how they look? Like with tiny little machines inside their
Yeah,
bodies?
I've seen those.
It's wild, right?
Right.
Well, imagine if we could actually do something like that, but for real. Not with robots and stuff, but by making super precise changes to these tiny little instructions that control our bodies, our genes.
Oh, wow.
That's kind of what we're diving deep into today. We're going to talk about CRISPR-Cas9. And it's way cooler and more real than any sci-fi movie out there, I promise you.
I mean, that's the perfect way to put it, you
Yeah.
know? This is like real life science fiction.
Yeah.
And we're going deep into it today.
Exactly. We've been looking at a whole bunch of scientific papers all about this CRISPR thing, how it works, what amazing things it can already do, and how it's changing medicine as we speak. It's mind-blowing. So our mission today is basically to try and help everyone understand this whole CRISPR thing, how scientists even figured it out, and why it's such a huge deal for treating so many diseases. We're going to explain it all in a way that everyone can follow.
Like we're just chatting with, you know.
Exactly. So you might be thinking, okay, what exactly is this CRISPR-Cas9 thing anyway? It sounds super complicated.
Right.
It's not as bad as it sounds, though.
Not at all.
Imagine you have this really, really long instruction manual, like millions of pages long.
Okay.
And it tells your body exactly how to grow, how to work, everything. And that's kind of like your DNA, right?
Right.
Now imagine you need to change just one tiny word in that manual or fix a single typo. How would you even go about doing that?
Oh, man.
It would be almost
I think
impossible.
a million pages. Forget about it.
Well, that's where CRISPR comes in. CRISPR-Cas9 is like this incredibly precise pair of molecular scissors, and they can find that exact typo and just snip it right out. It's like having a super accurate word processor for your DNA.
Yeah, it's super precise. And that's what makes it so revolutionary. It can go to a specific spot in your DNA and make a cut right there.
Okay, I'm getting the scissors part. But how do these scissors know where to cut? It's not like they can just read the whole instruction manual right.
That is a great question. The whole secret to CRISPR's precision is that it has two main parts that work together. So first you've got this enzyme called Cas9.
Cas9, okay.
Now you can think of Cas9 as like the blade of those scissors, the part that does the actual cutting.
Okay.
And get this, the coolest thing about Cas9 is where it originally came from. Are you ready for this?
Everybody hit me.
Bacteria.
Nuts.
Yeah, bacteria. It's part of their natural defense system against viruses.
Oh, way.
It's true. Imagine these tiny little germs are trying to sneak into your body, make you sick, right? Well, bacteria have their own version of that whole fight going on all the time. And they use Cas9 to find and chop up the virus's DNA, so they just totally destroy the virus.
So Cas9 is like a bodyguard for bacteria. That's
How-
so cool.
I know, right?
So, okay, that's Cas9, the blade, the bodyguard. What's the other part? What tells Cas9 where to go?
That's where the guide RNA comes in. We call it gRNA for short.
gRNA.
Got it.
And you can think of the gRNA as the GPS for the Cas9 scissors. Basically, it's a small piece of genetic material that scientists can design to match a specific part of the DNA.
Like a custom-made address.
Precisely. And this gRNA attaches itself to the Cas9 and basically guides it to the exact spot in the DNA, like programming a GPS to take you to a very specific address in a huge city.
That makes so much sense. So scientists create this guide RNA, and it's like the ultimate address tag, leading Cas9 right to where it needs to go. It's incredible.
Right.
So when did scientists actually figure out how to use this bacterial bodyguard system to edit genes and other living things? It seems so futuristic.
I know. The big breakthrough that got everyone super excited about CRISPR was back in 2012. These two amazing scientists, Jennifer Doudna and Emmanuel Charpentier, they discovered that they could actually take the whole CRISPR-Cas9 system from bacteria and make it work in other living cells, even human cells. Wow. 2012, that's not that long ago.
Oh.
So before CRISPR, how did scientists try to edit genes? Was it like trying to fix a computer using a sticky note and a pen?
That's actually a pretty good analogy. Before CRISPR, we had other gene editing tools. Things like zinc finger nucleases, we call those ZFNs, and talons. And yeah, they also aimed to cut DNA at specific locations, but designing and using them was so much more complicated.
Oh man, I bet.
Like, imagine trying to find a single word in a giant dictionary, but you can only flip through pages randomly.
Yeah, good luck with that.
It would take forever. You might not even find the word you're looking for.
So basically, ZFNs and talons were like old-fashioned paper maps.
Mm-hmm.
And then CRISPR came along, and it's like suddenly everyone has a smartphone with Google Maps.
That is a perfect way to put it. CRISPR is so much simpler, so much faster, and way more versatile. Scientists finally had this easier and more reliable way to target and modify DNA. And it's really sped up research. We've been able to do things we could only dream of before.
That's amazing. Okay, so we've got the basics of what CRISPR-Cas9 is, but let's dive a bit deeper into how it actually works inside a cell. You mentioned that the gRNA guides Cas9 to the target DNA, right?
Yes.
What happens when it gets there?
So the gRNA leads the Cas9 to that precise DNA sequence, and then the Cas9 does its job. It cuts both strands of the DNA right at that spot. We call this a double-strand break. Like, imagine snipping a piece of string clean through.
Okay, so the DNA is cut, but doesn't that sound kind of dangerous?
Well, you gotta remember, the cell is smarter than we give it credit for. It has its own built-in emergency repair system specifically for dealing with broken DNA.
Oh, so it's prepared.
Totally. It's like having a tiny little repair crew in each of your cells that's just standing by ready to fix any damage.
I like that analogy. So the scientists can actually use these repair systems to their advantage.
Exactly. They can actually guide the repair process to make the changes they want to the genes.
Wow. So it's like the cell's trying to fix the cut and the scientists are kind of whispering in its ear like, hey, why don't you try this?
Basically. It's super clever. There are actually two main ways that the cell tries to fix a double-strand break. One is called non-homologous end joining. It's a mouthful, I know.
Non-homologous end joining, got it.
But basically, you can think of this as the cell just grabbing the two broken ends of the DNA and sticking them back together as quickly as possible. Like if you ripped a piece of paper and just grabbed some tape and stuck it back together without really caring if the two pieces line up perfectly.
That's the quick and dirty method.
Exactly. And this quick fix often leads to small errors, like adding or deleting a letter or two at the cut site. And if this happens in the middle of a gene, it can totally mess up the instructions, basically turning the gene off or disabling it. It's like if you change a word in a sentence and now the whole sentence makes no sense.
So if scientists want to turn off a gene, they can just let the cell's quick fix system do its thing and make a little mistake.
Exactly. It's surprisingly simple.
Now, what's the other repair method?
That one's called homology-directed repair. And this is where the cell's a bit more careful, a bit more precise, you know? Okay.
If there's a template available, like a spare copy of the correct DNA sequence nearby, the cell will use that as a guide to fix the broken strand.
Yeah.
Think of it like having the original instructions when you're trying to fix something.
Oh, so it knows what it should look like.
Right. And here's the really cool part. Scientists can actually provide the cell with a custom-designed DNA template, one that has the exact change they want to make.
Oh, wow.
It's like giving the cell a cheat sheet.
I love it.
So the cell uses this template, repairs the break, and boom, it incorporates the new genetic information. And this allows scientists to do all sorts of amazing things, like inserting new genes, correcting faulty ones, or even just making very specific changes to the existing DNA.
So it's like they're giving the cell the perfect instruction manual and saying, here, fix it like this. That is so clever. Okay, so we've been hearing a lot about CRISPR being used with these things called organoids.
Oh yeah.
What are those exactly, and how are they helping scientists study diseases? So organoids are pretty fascinating. Imagine you could grow tiny 3D versions of your organs in a lab, like mini organs in a dish.
Oh.
That's essentially what organoids are. They're small clusters of cells that scientists grow in a special way so that they organize themselves to kind of mimic the structure and function of a real organ, like your liver, your brain, your intestines. It's like creating a tiny model of the real thing.
Okay, so mini organs in a dish. That already sounds like something out of a sci-fi movie. Why are these things so useful for studying diseases, and how does CRISPR fit into all of this?
Well, the great thing about organoids is that they behave a lot like the real organs in your body, but they're much easier to study because they're right there in a dish. You can poke and prod them and see what happens. So let's say you want to understand a disease like cystic fibrosis.
Okay.
Now, cystic fibrosis is caused by a problem in a specific gene, and scientists can use CRISPR to actually introduce that exact same genetic problem into a healthy lung organoid that they've grown in the lab.
So they're basically recreating the disease in a dish.
Exactly. They now have a model of the disease right there in front of them that they can study up close.
Wow.
It's amazing.
So instead of trying to study the disease in a whole person or an animal, they can study it in this tiny lab-grown version. That's a game changer.
Oh, absolutely. And they can do the reverse, too.
What do you mean?
Well, they've taken organoids that were grown from patients with cystic fibrosis and used CRISPR to actually fix the faulty gene in those organoids.
Really?
Yeah, and this really shows us the potential of CRISPR for actually correcting the root cause of a disease, at least in the lab.
That's incredible. So they can create a model of the disease and then test potential fixes all in the same mini organ. It's like a one-stop shop for disease research.
Yeah.
Our sources also mentioned something about using CRISPR to figure out how multiple genetic changes can lead to diseases like cancer.
Oh, yeah, that's another really important area of research.
How does that work?
So sometimes getting a disease like cancer isn't just about one thing going wrong. It can be more like a series of small problems that build up over time. And each of these problems makes the cell a little bit more likely to become cancerous.
Okay, following.
So this is often called the multi-hit model. Like, getting hit multiple times can really knock you down.
Yeah, that makes sense.
And the cool thing is that CRISPR allows scientists to actually mimic this whole process in the lab. They can take healthy organoids, like colon organoids, for example, and use CRISPR to introduce specific genetic changes that are known to be involved in colon cancer. So maybe one change makes the cells grow a bit faster, and another change makes them ignore signals to stop growing.
Oh, sounds like trouble.
And by introducing these changes one by one, or even in different combinations, they can see how the organoids gradually develop cancerous features. It's like building a tower block by adding bricks one at a time and then seeing how each brick contributes to the final structure.
So they can actually recreate the step-by-step process of cancer developing in a dish.
Wow. That's got to be so helpful for understanding how cancer progresses.
Totally. And hopefully it'll lead to new ways to stop it from happening in the first place.
So we've talked about how CRISPR is being used to study diseases. But what about actually treating people?
Ah, yes, the big
What
quiz.
kind of potential does CRISPR have for treating diseases?
This is where things get really exciting because we're talking about actually fixing the problem at its source. So imagine being able to treat genetic disorders by going in and correcting the faulty gene. That's the dream.
Like a permanent fix.
Exactly.
So we talked about fixing the cystic fibrosis gene in organoids. Is that a step towards actually developing therapies for people with cystic fibrosis?
It absolutely is. And just imagine if you had a disease that was caused by a typo in your body's instruction manual.
Right.
And the doctors could use CRISPR to go in and fix that typo permanently. Wouldn't that be amazing?
It would be like getting a software update for your body. Fix the bug and you're good to go. Exactly.
I also remember reading about CRISPR being used to modify immune cells to help fight cancer.
Oh, yes. That's another really promising area of research. It's called CAR T-cell therapy.
CAR T-cell therapy.
Yeah. And you might have heard of it. It's pretty cutting edge.
I think so.
So basically, scientists take a patient's own immune cells called T cells. These are the cells that are supposed to be fighting off infections and diseases.
Right, the good guys.
Exactly. And then they modify these T cells in the lab to make them even better at recognizing and attacking cancer cells.
Oh, wow.
And CRISPR can actually make this whole process way more effective.
Really? How?
Well, for example, they can use CRISPR to knock out certain genes in these engineered T cells that might be stopping them from working as well as they could against the cancer.
So it's like giving your immune system a boost.
Exactly. It's like giving your body's own defenders a superpower to fight the cancer.
Like a superhero upgrade. Amazing. Okay, what about the potential for actually curing inherited diseases?
That's the ultimate goal, right?
Yeah.
To develop a one-time treatment that can correct a genetic mutation for good. And then the person is essentially cured. We're not quite there yet, but there's been some really encouraging research in animals.
Like what?
For instance, scientists have used CRISPR to correct the genetic defect in mice that have a disease similar to Duchenne muscular dystrophy.
I've heard of that. It's a pretty serious muscle disease, right?
It is. And while this is still early stage research, the potential is huge. We might be able to develop these kinds of curative therapies for many different inherited diseases in the future.
It's mind-blowing. It feels like we're living in the future already. But this all sounds so complex.
Oh, it is.
Is there anything that can help make CRISPR even better, even more precise and safer? Because I know there are always risks involved with this kind of stuff.
Yeah, that's a good point. You're right. We have to be super careful. But that's where artificial intelligence comes in. AI. It's like having a super smart assistant for CRISPR research.
AI. Interesting.
Remember the guide RNA?
Yeah, the GPS for the Cas9.
Exactly. Well, AI can help scientists design the best possible guide RNAs because we want to make sure that CRISPR is super accurate. We don't want any accidental cuts in the wrong places. Those are called off-target effects.
And those are bad.
Not good, no. So AI is helping us minimize those risks.
So AI is like having this super accurate map maker to make sure the GPS sends those molecular scissors to the exact right address.
Precisely. AI tools can analyze mountains of genetic information and actually predict how well a specific guide RNA will work and how likely it is to cause any off-target cuts.
It's like having a weather forecast for your gene editing experiment.
I like that. It is. You can see which guide RNAs are most likely to be successful and safe to use.
It's incredible. It's like having this super smart assistant who's double-checking all your work to make sure you don't make any mistakes when you're editing the genetic code.
Exactly. And AI can do even more than that.
Really?
It can actually help scientists figure out which genes are the best ones to target in the first place for treating a specific disease. It can analyze tons of data and find those key players. And it's not just for the original CRISPR-Cas9 system either. AI is being used to improve these newer, even more precise gene editing techniques, like base editing and prime editing, which are like even more refined versions of the CRISPR scissors. It's like AI is helping us invent even better tools.
So the CRISPR is this amazing tool, and AI is helping us use it even better.
Absolutely. It's a really exciting partnership.
It is. Okay, so let's try to sum things up for everyone listening. We've covered a lot.
We have.
So the main takeaway is that CRISPR-Cas9 is this incredibly powerful and precise tool for editing genes, like these super tiny molecular scissors that are guided by a GPS.
A very, very specific GPS.
Right. And the whole idea was inspired by a natural defense system that bacteria use.
That blows my mind every time.
Me too. And now we're using AI to make it even better and even safer.
It's remarkable, and this whole technology has the potential to change how we understand and treat all kinds of diseases, from genetic disorders to really complex illnesses like cancer.
It's pretty amazing to think that scientists can now make such precise changes to our genetic code. The future of medicine is looking pretty wild.
It is. It makes you think about all the possibilities, but also the responsibilities that come with this kind of power.
That's a great point.
And if anyone wants to explore this topic more, there are some great resources online. Just search for CRISPR gene editing or AI in medicine.
Absolutely. Thank you all for joining us on this deep dive into the world of CRISPR-Cas9. It's a truly revolutionary area of science.
And we're just getting started.
Exactly. Who knows what incredible breakthroughs are just around the corner?
I can't wait to find out.
Me neither. Thanks again, everyone.
Thanks for listening.